Hello, AP biologists. Welcome to BioCast number 4. Hopefully you just finished listening to, taking notes about, thinking deeply about viruses. Now we're going to move up a level of complexity and talk about living cells. Okay, another quick reminder. As they were before, the slides in this podcast are available as a notes handout PDF file on the AP Biology page of mrtraphagen.com. If you have a printer and haven't printed them out yet, please pause the podcast and print these before moving forward. I'll join you on slide 3. Okay, you've seen this figure before, but now we're talking about the yellow flagellated bacterial cell. Bacteria are prokaryotic cells, meaning they lack a nucleus and other membrane-bound organelles. They're the smallest, simplest thing that we consider to be alive. So they're small, but not nearly so small as viruses. A typical bacteria, if there is such a thing, is approximately 1.0 micrometers, or 1/1,000,000th of a meter across. Bacteria, like viruses, are constantly evolving. This is why they continue to make us sick. As soon as our immune system learns to fight off one kind of bacteria, we then get infected with something else. Okay, more details on bacteria. As I said, bacteria are prokaryotes. They reproduce by mitosis. You take this single cell, you duplicate the DNA, you split the cell, you have 2 identical daughter cells. In bacteria, we call this binary fission, or splitting into 2. The other thing we need to know about bacteria is they grow very, very rapidly. Every 20 minutes, one bacterial cell can reproduce to make 2. Which means after 20 minutes, one cell makes 2. After 40 minutes, 2 make 4. After an hour, those 4 have made 8, etc., etc., etc. If these weren't limited by space or by food, these could cover the surface of the Earth in 2 days' time to the height of 1 meter. As it is, if we grow these in liquid culture, you can take one bacterial cell and make 100 million overnight. Bacteria are the dominant form of life on Earth. Far and away, these are the most numerous living things on Earth, both in terms of diversity and in terms of number of individuals. I'm sure you've heard me say by now, there are 10 times as many bacteria and viruses living on or in your body as there are people living on the Earth, which means you have 60 billion bacteria and viruses living on your body, and most of that 60 billion number is bacteria. This huge number and short generation time leads to an incredible amount of diversity, which you're going to see in picture on slide 5. Okay, a brief note on bacterial diversity. Basically, bacteria can be categorized into 3 different kinds of shapes. They can either be rod-shaped, like the green bacillus bacteria on the top left. They can be cone-shaped, or they can be sphere-shaped. Okay? So, some famous bacteria maybe that you've seen. The top right, the red guys, are Escherichia coli, or E. coli. We use them a lot in research. You will use some safe E. coli in one of our labs this year. Also, E. coli can be dangerous. They can make you sick. Very common form of food poisoning. Speaking of food poisoning, a cone-shaped bacteria, Salmonella. This is why we cook our food. Salmonella is at the bottom right of this screen. Moving immediately to the left of Salmonella, we see Streptococcus. Streptococcus is a family of bacteria that includes the bacteria that gives you strep throat, strep being a shortened version of Streptococcus. Streptococcus also includes Streptococcus pneumoniae, which causes pneumonia. Remember, that's the bacteria that Griffith used in his experiment where he made mice sick by taking heat-killed bacteria and safe bacteria and mixing them. He discovered that transforming factor, which we're going to talk about in just a minute. Okay, so prokaryotes are simple little critters. They have a haploid genome, which means they only have one copy of each gene. If that gene breaks, that bacterial cell is probably going to die. They don't have a nucleus. We call the naked DNA that sits in the middle of their cytoplasm, we call it the nucleoid. But basically, you just have that DNA hanging out in the cytoplasm. So, transcription from DNA to RNA and translation from RNA to protein happen one right after the other. You transcribe a piece of RNA, it doesn't have to leave a nucleus or anything, it just goes right to the ribosome and gets translated into protein. Prokaryote genomes are very, very small. They are only about 4 million base pairs, or 1/1000th the size of a eukaryotic genome like ours. And these are very simple little organisms. 4 million base pairs works out to about 4,500 genes. So these, we say they're very simple, very small. We're still talking over 4,000 genes for the simplest life on Earth. Okay, so we said bacteria reproduce by binary fission, which is mitosis in a bacterial cell. So, as you know, in order to do mitosis, you're going to have to replicate your DNA. Well, bacteria have one circular chromosome. Okay, that circular chromosome, where do you find a place to start? Okay, there is a piece of DNA that the bacteria knows to use to start. That's called the origin of replication. And if you look at the spherical piece of DNA on the left side of the screen, that little gray box is the origin of replication. Okay, now you then get replication in both directions, both clockwise and counterclockwise from the origin of replication, and we get a termination of replication site exactly halfway around that circle. Okay, that circular chromosome is being duplicated to make two interlocking circles. When you get to the termination of replication, they separate, and then you can do anaphase and telophase in bacteria just as you would in eukaryotes. Okay, we're going to pause for a thought question now. Bacteria reproduce asexually. Remember, mitosis produces identical daughter cells, so they're producing only clones of themselves. If that's true, where does this massive genetic variation come from? You saw in the picture slide back on slide 5, all of these different kinds of bacteria, and we know our immune system can learn to kill things it's seen before, yet we still keep getting sick. So you know there's a lot of genetic variation in bacteria. Where do you think it comes from? Don't look ahead, write what you think, and I'll join you on slide 10. Okay, welcome to slide 10, and let's talk about the sources of variation in bacteria. First and foremost, these are simple, small, barely alive little creatures, so they get mutated a lot. They don't have as many DNA repair mechanisms as we do, so if they make an error in replicating their DNA, that mutation is going to stick with them. Also, because they're single-celled and they're small, they're exposed to lots of environment. So any sort of mutagenic chemical or environmental stress can cause more mutation in bacteria than we would be used to as big, strong eukaryotes. Next, there's a thing called conjugation, and I'm going to save that for the end, but it's kind of, sort of like bacterial sex. Okay, one bacterium can extend this thing called a pili to another, and they can exchange some genetic material. They're not really having sex, but they are exchanging material. And then there's this thing called transformation, which I'm going to talk about next. And transformation is when you can introduce new DNA into a bacteria, either on a thing called plasmid, which is a circular piece of DNA. You see it in that conjugation diagram, that F plasmid. It's a smaller circular piece of DNA. And you can also introduce small DNA fragments. Let's talk about transformation with plasmids first. Okay, bacteria are stupid. They will copy any DNA that shows up in their cytoplasm. Their transcription and translation processes don't know their own stuff from foreign stuff, unless, remember, it has those restriction sites like GGCC that I talked about in podcast number 3. We'll get back to restriction sites later. The point is, you can take a plasmid, a circular piece of DNA, put it into a bacterial cell, and it will reproduce that plasmid over and over and over again. How could we use this? Biotechnology, yay! Biotechnology is something we do now to use bacteria to do medical research, to do manufacturing, to do things we want them to do. Let's talk first about using bacteria as protein factories. So to do this, you take a gene that we're interested in, like let's say the gene that codes for the protein insulin, and you put it on a plasmid. If you can put that plasmid into bacterial cells, they will transcribe that DNA into RNA, they will translate that RNA into insulin protein, and remember, they're reproducing every 20 minutes. Okay, overnight, one cell can become 10 million cells. 100 million cells. So we're making a lot of these bacteria, and all of these bacteria are now making insulin. We can then harvest that insulin, put it in little bottles, and give it to people who have diabetes, who can't make their own insulin. How does that work? Okay, this is an example of a plasmid. Okay, this diagram, PUC18, okay, is a plasmid that has some genes we're interested in. And then if you see on the left side of the screen, you see that big orange arrow that says ampicillin. We put a gene on this plasmid that makes the bacteria immune to ampicillin. We call this an ampicillin resistance gene. On the lower right, you see ORI. That's the origin of replication. That shows you where replication of this plasmid is going to start. Okay. Then you'll see in the upper right side of the plasmid, you see all those abbreviations like HIND3, SPH1, PST1, all of that at the bottom of the list, ECHO R1. Those are all restriction sites. Those are all places our plasmid could be cut open by different restriction enzymes. And the restriction enzymes are always named for the bacteria we got them from. So for example, ECHO R1 comes from E. coli, and it's restriction site 1. We take our plasmid, using our knowledge of restriction sites, and we take, if you see in the upper right part of the screen, that little white rectangle, that's our gene of interest. Let's say that's the DNA that codes for insulin. We cut the plasmid open using one of our restriction enzymes that we stole from some other bacterial cell. We put our insulin DNA into the circular vector, and now we have a circular piece of DNA, a circular plasmid that includes the insulin protein. We put that plasmid into a bacterial cell, and now that bacterial cell will manufacture insulin for us. Okay, well, before I talk about transduction, pause the podcast. Make sure you've got your notes on transformation using plasmids to introduce new DNA. Okay, now let's talk about transduction. Now, this is something that happens out in the natural world all the time that we have stolen for biotechnology. Transduction, as opposed to transformation. Transduction is the introduction of new DNA into cells using viruses to deliver that DNA. So in this case, we are doing transduction. We are introducing new DNA into a bacterial cell using a phage virus. So if you start in the upper left corner of the diagram, you have a phage infecting this bacteria that has A+ and B+ on its chromosome. Now, this looks like it's a lytic virus because it's chopping up all of the stuff inside that bacterium. It's assembling new viruses, and as those phage viruses are assembled, some of them take up some of that A+ or B+, those genes from that first bacterium. One of those phage viruses infects a second bacterium that is A-, so it doesn't have whatever that A gene is. The virus introduces the A+ gene. So after infection by this virus, this second bacterium is now A+. That A plus has been introduced by the virus. Now, phage do that all the time, but medical researchers, including my wife, use these viruses to introduce DNA or RNA into cells that didn't have them before. This works with bacterial cells. It also works with human cells, although that's more complicated. We'll save that for a new podcast. Okay, I promised you I'd talk to you about bacterial sex, so here you go. Conjugation. Again, it's not really sexual reproduction, but it is an exchange of genetic material which leads to additional genetic diversity. This is a picture of conjugation. Now let me tell you about it a little bit. Okay, conjugation is the transfer of DNA between two bacterial cells. They're joined by this bridge, which is called a pili. Okay. Now, in order to do this, build this bridge, you have to have something called an F factor. We just called it that because F is the first letter in fertility. Okay. If one bacterial cell has an F plasmid with that F factor, it can make this pili. Okay. And so we've got to call it something. We'll call that the male bacterium. It extends this sex pili, attaches to a bacterium that doesn't have one. We'll call that the female. And then this pili contains a little bit of cytoplasm, and that allows DNA, like plasmids, to be transferred between the two bacteria. Now, this can cause some serious problems because since it isn't really sex, these two bacteria don't have to be of the same species or even in the same evolutionary group to exchange material. And if your plasmid has something like a gene for antibiotic resistance, it can be passed from any bacterial cell in the world to any other bacterial cell in the world. So, if by spontaneous mutation, some bacterial cell evolves the ability to beat ampicillin, that ampicillin resistance gene can be transferred to any bacterium it comes in contact with. This is one of the reasons bacteria evolve so quickly. They can transfer their genes without reproducing. This is why antibiotics are constantly becoming obsolete, because bacteria can evolve so quickly and transfer these beneficial plasmids between one another so quickly that we're never going to catch up with antibiotics. Not to scare you or anything. Okay, that brings us to the end of BioCast number 4. As always, make sure to write down any notes, questions, concerns, etc. while they're fresh in your mind. We will talk about this in class. Okay, coming tomorrow, not due for Monday, there's a special podcast 4.5, which talks about bacterial metabolism and the operon concept. The operon is really complicated, so we're going to split that off into its own podcast. Please read concept 18.4 so you've sort of heard of this operon stuff before you listen to the podcast. Having said all that, happy studying. I'll see you on Monday.